Yeah. No. Okay. Go. Today. All right, cool. Today we're going to be reviewing cellular metabolism. So we're going to be going through, I think, chapter 3 in your book. You should all have seen this at least once before in some other class. I want you to jog your memories of cellular metabolism right now before we talk about it together. So we're going to take 2 minutes to write down what you can remember. So you're thinking step of aerobic cellular respiration or glucose metabolism, number of different things you might have called it. We're going to be thinking about ATP production. So you want to be thinking about which steps produce ATP, and if you know how much, great, put that on there. So I'll give you 2 minutes to jog your memories, and then we'll fill in the gaps. This is also for you to remember what you might have forgotten. Hopefully by the end of the unit, this will be easy peasy. We'll be able to do it 2 minutes, no problem. Just for helping me know where we're at here. Who thinks they remember metabolism and cellular respiration like perfectly, didn't miss any steps, could do all this like that? Okay, great. It's worth me talking to you. I always wonder something I know you've seen sometime before. Right, perfect. So we're going to be talking today about how we break down ATP how we break down glucose and turn it into ATP, because ATP is going to be powering cellular reaction, cellular responses all over the place. Okay, so we're going to be describing the events in glucose metabolism, and they might be a little different than when you talked about them before. None of the actual process is different, just different textbooks kind of lump them together slightly differently sometimes. So we're going to be going through them as glycolysis. Glycolysis is always glycolysis. And we're going to jump to the Krebs cycle. So there's a linking step with pyruvate that is sometimes delineated out as a separate step. So we're going to jump directly into talking about the Krebs cycle, and then into oxidative phosphorylation using the electron transport chain. And then after we go through like normal cellular respiration, our aerobic cellular respiration. We'll talk about other ways we can get energy. We'll think about what we do if we don't have oxygen, which we're going to need for our standard typical breakdown of glucose. We're also going to think about, okay, what if we want to break down something that's not a carb? How do we break down fat? How do we break down protein? So that'll come at the end. We're going to start by thinking about ATP. We're going to test our memories of some chemistry words as well here, just so they don't trip us up if they show up in questions later on. I'm not directly going to be testing you about chemistry vocabulary in this class, but I want to make sure that you can actually answer the A&P questions and not miss them because you missed some word that we expect you got in a chemistry class. So when we describe something as endergonic, what do we mean by that? So we know it's about energy. We got a little bit of mix. Yeah, we're talking about energy of products and reactants. And in an endergonic reaction, we're thinking about a reaction where our product ends up with more energy than our reactant had in the first place. So last time, when we were thinking about activation energy and enzymes, right, we were thinking about reactants that had a certain amount of energy. So energy was here, reaction is here. They had reactants and activation energy and then products with lower energy, right? So that would be an exergonic reaction. But in an endergonic reaction, we have the opposite thing happening, right? So we have a product that has more energy than the reactants we put in, right? So we need a source of energy to put into this reaction to get more energy in those products. So endergonic, ender, I like to think of as meaning like inside. So it's got to pull energy in from somewhere. So the reason that we want to know that word endergonic, is that the formation of ATP is going to be an endergonic reaction, right? We're trying to trap energy in the ATP. So it's going to happen paired with a series of exergonic reactions, so reactions that release energy. So that we're releasing energy so that it can finally go into creating this final covalent bond in our ATP molecule. Okay. So we looked at ATP molecules as relates to nucleotide structure and nucleic acids last time. So you might remember that we got this adenosine structure. So we've got this kind of adenine nitrogenous base here. We got a ribose sugar and we got some phosphate groups. The reason this isn't just a nucleotide is we have more than we usually would. So ATP at T stands for 3, adenosine triphosphate, right? So that triphosphate, worst 3 in the world, that triphosphate is talking about these 3 phosphate groups here. Right? Those 3 phosphate groups there are in ATP. ATP is holding energy in the bonds between those phosphate groups, in the covalent bonds. And so when we make ATP, what we're doing is we're going to be starting with ADP, adenosine diphosphate, and we're going to be forming one additional bond here. I think it's that one. Ask a chemist about exactly which one it is. We're forming an additional bond to an additional phosphate group so that we now have three. We soak energy into that bond. And when we go to use ATP, this is helpful for us, right? So when we say something requires energy, we snap that last phosphate group off. When we snap it off, that energy that was in the bond gets released, and we can use it to like push something across the membrane, right? So that's why we're storing energy in the ATP. So making ATP is an endergonic reaction. Using ATP is an exergonic reaction. And in order to do an endergonic reaction, we got to get the energy from somewhere, which is why all the steps of metabolism are involved, right? We've got all these steps that we're setting up in order to actually get all this energy to put into the ATP. Okay. So here you can see the exact number on that, although I won't be asking you for that. So we can see that ATP synthesis, right? So synthesis means making something. So ATP synthesis means making ATP, right? It requires an additional 7 kilocalories per mole in order to form that ATP from our little reactants on there, right? Which are ADP. So that adenosine diphosphate. So that's the 2, that D in the middle. D is 2. Here's that third phosphate group that we're grabbing from the environment, right? So that's where the third one can come on so that we have our adenosine triphosphate at the end. But we need to put energy into this process. So this does count as a condensation reaction as well. So if you'll notice, we make some water as a byproduct here. We might also refer to it as a phosphorylation reaction. So these are all different things that are true about this process, right? Different ways we could describe it. A phosphorylation reaction, because we're talking about a phosphate group, right? So phosphorylation is referring to the fact that we're adding a phosphate group, right? So when we add a phosphate group, that's phosphorylation. And we do it in 2 different ways in this process of aerobic cellular respiration, right? So not only do we conceptually have 2 ways we can do it, we actually do it both ways in our bodies all the time. Okay. So one way we can phosphorylate something is substrate level phosphorylation. So last time we talked about enzymes and substrates, right? So substrates, remember, we're binding in on to enzymes, right? So we have that active site, substrate goes in there, and the enzyme catalyzes that reaction. So that's one way we can do this phosphorylation. We can do substrate level phosphorylation mediated by enzymes that work in that way, that induced fit model, similar to the A&P model. We can wave our hands a little bit. The other way that we can do phosphorylation, so the other way we can add on a phosphate group, is through oxidative phosphorylation. So in oxidative phosphorylation, we're thinking redox reactions, except we're not in chemistry. So we're going to remember that oxidative phosphorylation is really the step that's going to come at the end of aerobic cellular respiration. And that's the part where we need the electron transport chain to help us move electrons around and build up a gradient, and ultimately use that to build our ATP molecules. So this oxidative phosphorylation process looks different than our typical hold a substrate in place on an enzyme and pack things together. This is a more specialized process. This is where we're going to have a bunch of electron transport. Okay. So in metabolism, in this unit, we're going to be thinking mainly about making ATP, but we're going to be looking at ATP throughout this course. And the time when it's going to come back and be really important for us is muscles and how muscles work. So I picked a little picture that will become very familiar to you in the future after we talk about how muscle contractions occur. And you can see that there's ATP on it, right? That's why this picture is on here right now. We got ATP. Right, so we use ATP in different cellular processes. this is an example of one of those cellular processes, to release energy to actually have some effect in the body. So in this case, we're using ATP and breaking that ATP down to ADP in order to change the shape of some protein. Okay, I'm going to change that shape. Okay. And we'll see ATP being used lots of other places, like when we think about active transport in probably next week or in a couple of weeks. Okay. So ATP is a temporary way that we store energy. ATP is small, we can move it around the cell pretty easily, it can bind into all sorts of receptors easily. It's useful for us. And so when we break down ATP, we're just going to do the reverse of how we formed it. So we take our ATP, our adenosine triphosphate, with its 3 phosphate groups. We add in some water, and then that's going to break it apart into the adenosine diphosphate and an extra phosphate group, plus the energy that was in that bond that we've just broken. So this would count as a hydrolysis reaction because we broke something, right? We lysed it. That lysis tells us we're breaking something down. Hydro for water. So we added water and we broke the ATP. Hydrolysis reaction. So for the most part, I'm not going to be testing you on these exact numbers. We're going to be focusing on like the conceptual process of cellular respiration. They are on here so that we can piece together how it works, so that we can understand what's happening specifically here in class. And then we're going to make sure that we understand the fundamental concepts on the exam. So when you're thinking about like what numbers you want to know, definitely know numbers for like how many ATP are produced in glycolysis, but don't worry about like how many kilocalories are released by glucose oxidation. I'll cue you when there's a number that you definitely want to remember. Okay. But we are going to look at the whole reaction to be complete right now while we're talking about it first. Okay. So what we're doing in aerobic cellular respiration, so cellular respiration that uses oxygen, which might also be called glucose oxidation, as it is in this slide. We're taking glucose. So here's our glucose, our C6H12O6. We're going to need oxygen as part of this process. So here's our 6O2, our 6 oxygen molecules. These are usually not drawn in the equation for cellular respiration, but they are important. So we're going to need some precursor molecules for our ATP. We got our 38 ADP molecules and 38 phosphate ions. They're going to give us 38 ATP when they come together on the other side. And if we focus on what happened to that glucose and oxygen, we can see that in this process, they've broken down into carbon dioxide. So there were 6 carbons in our glucose molecule, so 6 carbon dioxide. We can breathe out carbon dioxide. This is why we breathe out carbon dioxide, right? You're eating carbs, you're breaking down the glucose. You breathe out those carbons that you don't need anymore because you moved all the energy into an ATP molecule. And we also have water as a byproduct. Lots of water in our bodies, so this might not be super surprising, but you can feel like how humid your breath is. So you can also think about how we're eliminating water as a byproduct of this process. If you did want to do out the math of this step, let's see that for those 38 ATP molecules that we're producing, there are approximately 266 kilocalories that we're going to need. Right, so we said it's an endergonic process, and that amount of energy we need is 7 kilocalories per mole. We're going to see that the energy released by this process of breaking down glucose, right, so this glucose oxidation, we really just mean breaking down glucose. Right? It's actually way more than we actually need, right? So we have 686 kilocalories of energy that we release when we break down the glucose molecule, but only 266 kilocalories that we need to make that ATP. So this is a difference of, if I did the math right before, 420 kilocalories, which means this is not the most efficient process, right? So we are actually only using about, well, under 40% of the energy in that glucose molecule, right? So we're not using all the energy we could be from breaking down the glucose molecule. Some of the remaining energy that we don't use just becomes body heat, right? So this is helpful for homeostasis as we're trying to maintain body temperature. Body heat is coming out of this reaction as some of that kind of excess. So this is not a perfect process. Not all our energy ends up in an ATP molecule. But not to make your body run, so that's nice. We're going to get down to the steps next. So here we have my favorite picture of glucose oxidation or aerobic cellular respiration. Okay, so this is something you might compare to your pictures that you were drawing or the steps you were listing. All right, so we have a process that starts with glucose. All right, so that's our C6H12O6, so our carbon-based molecule, our carbohydrate that we're breaking down, with our goal of making ATP. You'll see numbers of total ATP made by cellular respiration can vary a little bit. This is actually because, like, in different tissue types, it actually can be a little bit variable. Exactly how efficient that oxidative phosphorylation is isn't completely static. So don't get tripped up by exactly how many we see at the end. But we are going to see that in the earlier stages, we'll have consistent numbers. So glucose starts our process. We have glycolysis happening as our first step. In glycolysis, we break down the glucose. We basically chop it in two, and as we chop it in two, we release on net 2 molecules of ATP. Okay. And 2 reduced electron pairs, so our NADH here. You may or may not have put that in your little notes to yourself. I didn't ask you to talk about NADH, but we will eventually. Okay, so first step was glycolysis. We break the glucose in half. We release 2 ATP. Then our next step, we start with, this is our linking step in your textbook, might have been a separate step in your previous classes, but we take pyruvate. We're pulling that pyruvate from the cytoplasm into our mitochondria, and we're converting it into 2 acetyl-CoA molecules. Okay? And along the way, we're reducing some more electron carriers. So you see more NADH, and we get rid of some carbon, so that's some of our CO2 that we're breathing out, some of that back there. So you might have had that as a separate step. And then we have the next ATP producing step that we're going to focus on. I think this is why our textbook picks those 3 specific steps. If you think of glycolysis as a step, it produces ATP. You jump to the Krebs cycle as your next step. That's the next one that gives you some ATP. And then oxidative phosphorylation will give you ATP as well. So Krebs cycle happening inside our mitochondria produces some ATP. So we can see 2 ATP per molecule of glucose. We'll talk about why I'm specifying that in a little bit. Okay. And it's doing a lot of work when it comes to our reduced electron carriers here. So we see that we have a lot of NADH forming, a little bit of FADH too as well here. So that's going to be kind of the main job of our Krebs cycle. We'll see in a little bit. Finally, we're going to see our oxidative phosphorylation step. So that's the one where we have our electron transport chain, passing electrons through proteins embedded in a membrane in the mitochondria, building up a big gradient, a proton gradient, and ultimately giving us a ton of ATP. Okay. So we're just going to think of this as a lot, somewhere between like 20 and 30-ish. Okay. So glycolysis, Krebs cycle, and oxidative phosphorylation are our three steps that produce ATP. We got a lot of other things going on along the way. Now, since you've just mentioned our ways of phosphorylating molecules, and that we do it two ways in metabolism, point out in these steps that we compare specific steps to specific ways of phosphorylating. So substrate level phosphorylation is happening in glycolysis and in the Krebs cycle. So that's the one where we're using enzymes to like hold ADP and phosphate ions in place so that we form a bond between them. So that's what we see happening out in the cytoplasm in glycolysis and inside the matrix of a mitochondrion in the Krebs cycle. You might also have called this the TCA cycle, the citric acid cycle. Those are the same thing. I'm sorry. I didn't make these names up. Okay, so those are substrate level phosphorylation. And it's only at the end in oxidative phosphorylation that we have this new way of phosphorylating using the movement of electrons to help us tack on that extra phosphate ion to make our ATP. We're going to start with glycolysis. I said most of these things with my words, but now you got it in bullet points. Fundamentally, our goal when we are making ATP, the main way that your cells make ATP is by breaking down glucose. We'll talk in different organ systems, what cell types can use things other than glucose, but whether it's not all of them, like nerves and brain definitely kind of need to use glucose. So we're going to start with glycolysis, glucose breakdown. Okay. So our glucose is a 6-carbon molecule that by the end of glycolysis turns into a 3-carbon molecule, but we're going to end up with 2 copies. So we go from glucose to 2 pyruvate molecules. So there's a series of 10 steps in between, right? So we have 10 intermediate steps, are all enzyme catalyzed reactions. So you'd have lots of enzymes helping us do each of those 10 steps of glycolysis. This is all happening out in the cytosol, in the cytoplasm. So since I've just said that, I'm going to give you guys a reminder of mitochondria as well and cells. Okay, so when I'm pointing out that this is happening in the cytosol, okay, so if we have a cell, my big cell, okay, in general, that watery environment inside of it, we know is our intracellular fluid, our ICF, right? But it's also got a bunch of other stuff dissolved in it. We've got organelles and stuff floating around in it. When we refer to this all as one, we're going to refer to it as a cytosol, this kind of gel-like stuff here. Okay. So cytosol is just this kind of general environment we're working with. And when we're thinking about cellular respiration and metabolism, we want to distinguish what can happen in the cytosol, so just generally around about in the cell, versus what happens in the mitochondria. So I'm going to do a mitochondrion, but it's going to be bigger than a real one. Okay. So they look kind of kidney beans. Okay. So they got that one outer membrane. That's the line I just drew, the outer membrane of mitochondria. It's enormous mitochondrion. They got an outer membrane, then you got a little space, and then you got a foldy inner membrane. Yeah. So a lot of our steps, basically everything except glycolysis, when I say it's happening in the mitochondria, we're going to be thinking of it happening mostly in the mitochondrial matrix. I'm going to write matrix in here. Tablets do this. Okay. So that's our mitochondrial matrix. This whole structure here is our mitochondrion. And I'll finish labeling for us, I guess. So this outer one, right? Outer membrane. Then we have an intermembrane space, right? So this is a gap, right? Inter, be careful about that T, right? It's inter, not inner, because inner would almost be referring to the matrix. Okay, so inter, between, membrane space. Okay. Then we have the inner membrane itself, right? So now no T, because we're just talking about that inner membrane. And then the mitochondrial matrix in the middle. We don't need all those pieces for right now, but we're going to need them for the other parts of the process. So it's worth remembering why we're specifying that this is happening in the cytosol. Right, so glycolysis can happen out here. It's actually the only one of the steps that can happen out here. Glycolysis. That's happening out in the cytosol. Our other energy producing steps are going to be happening in the mitochondria. Okay. So on that, in the process of glycolysis, we are going to gain 2 ATP molecules. So it's important to note that this is a net gain, right? So net means like the total. The reason we're specifying is that if you look at those 10 different steps, we have to put a little bit of ATP in to kind of jumpstart the process. And then we make ATP, get the net number, we have to do some subtraction. So we'll see that on the slide in a second. But when I ask you how much ATP is made in glycolysis, this is the number. You want to know that glycolysis makes you on net 2 ATP molecules. That's important. We also reduce some NAD molecules. Okay. So I like to remember that sentence way back from when I was in high school chemistry to remember this. Leo the lion says Gur loses an electron. So this is for our redox reactions when we're thinking about what's oxidized. Okay. Sorry, guys. Okay. When we're thinking about why I'm remembering this, in case you haven't seen this acronym before, we're trying to make sure that we remember what oxidation is versus reduction, right? So this is reduced. Oxidized. Okay. So Le is losing an electron. And this Ge at the beginning of this Gur is gains an electron. The reason I'm reminding you that is that's how I remember that we have this reduced form, this NADH. So when we tack on this hydrogen, we're also tacking on electrons here. So we're gaining electrons, making this reduced electron carrier, NADH, as part of glycolysis. So this is going to be more important in our Krebs cycle, but it's also happening here in glycolysis. So other things to note about glycolysis is that nowhere in this equation should you see O2. We don't need oxygen for this process. Part of our whole big long workflow, if we're trying to get to the steps that need oxygen, but glycolysis itself doesn't require oxygen. And that's going to be important when we start to think about what can you do for energy if you're in a low oxygen environment. We're also going to notice that we're not actually losing any carbon yet, so no CO2 is being produced yet in metabolism if we're just in glycolysis. I got a full equation down on the bottom. So remember, as we're going through each steps, it's my reminder to remind you, when you're going through and trying to explain them to yourself, you want to be thinking about, okay, what is the name of the molecule that we're interested in? So think glycolysis, we're thinking we're going from glucose to pyruvate, right? Those would be what I mean by like, what's the name of the molecules? How much ATP we're using or creating, and this glycolysis step, you need 2 ATP on net. And then when we're thinking about that, reducing and oxidizing our electron carriers, we just saw that we made this reduced electron carrier, this NADH. And we'll come back to that as we keep going through. Make sure I told you. All right, so looking at that NAD and NADH, right, these are energy transferring nucleotides. So kind of think of them as like little cars that are carrying electrons to the end of the process, right? When that H hops in, it's hopping in with electrons, right? And we're going to be carrying them to our oxidative phosphorylation step inside our mitochondria. So these are count as something that works alongside our enzymes. So when we were talking about cofactors and coenzymes the other day, this is an example, something that's not an enzyme, but we're going to need to use it in conjunction with our enzymes at the end of this process to make ATP. Now remember that reduced, and we say we have a reduced form, that means it has an extra electron. We can think about the electrons, we can think about the chemistry, but the way I cheat on this is that I always think about that hydrogen, right? The hydrogen is carrying electrons with it, so I just think it gained a hydrogen, it gained an electron. So glycolysis is creating 2 of those. All right. So let's remember what we said a couple minutes ago. So when we were thinking about mechanisms of ATP generation, those 2 ATP molecules that we just made, that we synthesized, what was the term for that type of ATP generation? Okay, great. Yeah, so glycolysis, those 2 ATP we're making are through substrate level phosphorylation. So we'd be using enzymes to put our substrate into position in order to tack on our extra phosphate group. So here are all those enzymes that we're not going to be learning the names of in this course, but we can see in general how the steps of glycolysis work. So this is just that same process with a little more detail added. Right? So we can see that we go from glucose to pyruvate. We have a bunch of intermediate steps. Each of these intermediate steps is regulated by an enzyme. That's why we were talking last time about enzyme regulation. We want to have the appropriate amount of all these intermediates so we can just be smoothly going through glycolysis all the time. So we might have to slow down some steps, we might have to speed up other steps to make sure we have what we need. Okay, so here's what's happening there. And we could also see that if we wanted to break down glycolysis into kind of 2 conceptual steps, we've got an energy investment phase, and then we've got an energy harvesting phase. Okay. What I mean by that is that this first part of glycolysis, so where we go from glucose to glucose 6-phosphate to fructose 6-phosphate to fructose 1,6-diphosphate. Okay, so this was all before we actually broke anything in half, right? We didn't break glucose in half yet. So what we're kind of doing here is we're destabilizing the glucose so that we can break its bonds better. And to do that, we had to add in some ATP, right? So we start by investing some energy in this process. So here you can see we put in an ATP molecule and we use that energy to turn the glucose into glucose 6-phosphate. And then we did it again here. So in this part where we turned fructose 6-phosphate into fructose 1,6-biphosphate, that took a unit of ATP as well, right? And that puts it into this form that we can then start to break down. Right? So our energy investment phase costs 2 ATP to like set us up to do the rest of glycolysis, which is the energy harvesting phase. So here, if we zoom in on what's going on with that ATP, right, we can see now we're taking ADP and making ATP, right? So here we see we make an ATP. Here we make a molecule of ATP. We can see the enzymes involved, the phosphoglycerate kinase and the pyruvate kinase here. And on the other side, this would be happening exactly the same way. So we multiply this by 2. That's why there's a 2 in parentheses, because this is the point where we split it in half. So that's why we say that we have on net 2 ATP produced by glycolysis, because we put in 2 and get out 4, right? So you can think of it like it cost us 2 ATP, so we're gonna like make that a minus sign. If this helps, listen to me. If it doesn't help, ignore I ever said what I'm about to say, right? So think like you had to pay 2 ATP, sad, so a minus sign, but then you earn 4 ATP at the end, right? Which is how we get our 2 net, right? So negative 2 plus 4 is the same thing as 4 minus 2. So that's our 2 net ATP. The next piece of our puzzle is our linking step with pyruvate. So this is where we go from glycolysis, happening out in the cytosol, but our Krebs cycle is going to be happening inside the mitochondria in the mitochondrial matrix. So we need to actually get there, right? So that's happening in this linking step. We also have some transformation of the molecules involved, right? So here we can see our pyruvate coming in. And here we see coenzyme A, CoA, working along with pyruvate dehydrogenase, another enzyme involved in this process, giving us acetyl-CoA, which is the molecule that we're going to be using in the Krebs cycle in the next part of this process. Okay. So this is important, but it doesn't generate any ATP. It does generate a tiny amount of that reduced electron carrier. So we can see a little NADH in this process here. This would be another example of a coenzyme right here. This is acetyl-CoA working alongside our enzyme. That brings us to the Krebs cycle. It's also called the citric acid cycle, which is also abbreviated to the TCA cycle. Too many people were studying this at the same time. All the same thing. Okay. So we are now inside the mitochondrion. So I'm going to draw it again. All right. So we have our kidney-shaped mitochondrion. Mitochondrion is singular, mitochondria plural. Here is our inner membrane, right? So we had outer membrane, intermembrane space, inner membrane, and then matrix inside. Okay. And I'm gonna start at matrix because that's where the Krebs cycle is happening. We are in the mitochondria, not out in the cytosol, and specifically we're in the middle of it. So this is where we passed our acetyl-CoA from that linking step with pyruvate. And we then enter a cyclical process, which we mean it just keeps going and going and going around in a circle. We keep adding acetyl-CoA like to this Ferris wheel, but the Krebs cycle or the citric acid cycle is kind of always turning. So this process where we enter, this is going to begin where we add acetyl-CoA in and we make oxaloacetate. That's going to begin it, and then we're going to get the oxaloacetate out at the end of this process as well. And we're going to just keep circling around. It's happening in the mitochondria. It does produce ATP, although it's going to be kind of like the least important way that we generate ATP in our bodies. It does do it. So per turn of the Krebs cycle, so per like cycle, we make one ATP. But it's important to note, we break glucose in half, right? So for every molecule of glucose, we do this twice. Right? So that's why the numbers, you have to be careful what you're paying attention to, right? So per molecule of glucose, we make 2 ATP. But if we're just focusing on one cycle of the Krebs cycle, it's only going to show you one on the picture. The big thing that's happening here, though, the more important thing that's happening is the reduction of our coenzymes. So we are taking in a lot of NAD and also FAD and reducing them, so tacking on electrons to our shuttle, turning them into NADH and FADH2. So this is where most of these electrons that we're going to see entering the electron transport chain at the end of the process, this is mostly where they're coming from. We got a little bit in glycolysis and a little bit in that linking step with pyruvate, but most of it's coming from the Krebs cycle. Okay. So, big point of this is producing those reduced electron carriers, but we do make 2 ATP per molecule of glucose or 1 ATP per molecule of acetyl-CoA, depending on where you're starting your examination of this process. If you draw it in a flowchart form, it'll make sense how those two things pair together. And here's another big picture of it. So finally, we have our oxidative phosphorylation step. So coming into oxidative phosphorylation, so what we have just done today, we thought of glycolysis and we thought of the Krebs cycle, right? So if we're thinking per molecule of glucose for this first bullet point, per glucose. Right? So those first 2 parts of cellular metabolism were glycolysis and the Krebs cycle. We've only made 4 ATP molecules so far. So we saw in the original equation that we can make up to like 38 molecules of ATP by breaking down glucose. So we clearly are not all the way there yet. And it's this oxidative phosphorylation step at the end that's going to make the bulk of ATP. So tons of ATP, and this is the part where we're going to need oxygen. So we'll pick up with oxidative phosphorylation on Monday. And so don't forget to do your little review quiz based on the things that we've done in class this week. And